It's now with my great pleasure to introduce Trevor Pitcher, Dr. Trevor Pitcher. For today's webinar, Trevor got his bachelor's and master's in biology from York University in Toronto. He then got his PhD in zoology from the University of Toronto. He has been a professor at Windsor University since I think around 2006 at the Great Lakes Institute for Environmental Research. He's also the director of the Freshwater Restoration Ecology Center based on the Detroit River near Windsor. And I just learned that he is also the editor-in-chief of Canadian Journal on Fisheries and Aquatic Sciences, which is a great honor. Trevor has a really broad research program spanning evolutionary biology, reproductive ecology, and conservation biology. That work spans both coasts of North America and also includes the Great Lakes. His focal species include those that are sort of species at risk, like red-side dace, lake sturgeon, and those that support key fisheries, like Chinook salmon and lake trout. I've known about Trevor for a while, and especially some of his research on bloater broodstock, trying to enhance bloater broodstock production in the Great Lakes. But I had a chance to interact with him recently and learn more about some of the other research that he's doing. Again, some of it not Great Lakes, but all of it really relevant to Great Lakes. And the part that really was exciting to me is his research trying to improve the overall fitness of artificially reared fishes. And so from investigating whether enrichment in stream tanks can improve gametic quality, or how we can reduce transport stress, or what is the best stocking release strategy. Like Trevor's doing all that stuff with his students. So I was really pleased he accepted our invitation to join today. And this title, as you see here, is Using Reintroduction. Just blocked it there. Biology to Restore Freshwater Biodiversity in the Great Lakes Basin. So thanks again, Trevor.
Yeah, thanks for having me, Bo, and thanks for the great introduction. I do want to preface this talk by saying that, of course, all the work I'm going to speak about today, as you mentioned, Bo, is really a tribute to the graduate students and postdoctoral fellows that have come out of the lab. So I won't have a slide with all their specific names. They know who they are. A lot of you have worked with them. But I also want to acknowledge the agencies I'm lucky enough to work with. So I've had the opportunity to work a lot with Ontario Fish Culture, the Ministry of Natural Resources in Ontario, but I've also been fortunate to work with U.S. Fish and Wildlife and even USGS. So a lot of this work that I'm going to talk about today wouldn't be possible with any of those people and agencies. So I just want to acknowledge them up front in a sincere way and tell you that I'll finish the talk with some hot takes on how we can maybe improve fitness, but I can tell you there's nowhere I'd rather be than either on the river or in a hatchery. So without further ado, I guess I'll just tell you that, you mentioned the title, we're really focused these days on reintroduction biology. We're working on this in British Columbia, in Nova Scotia, but also here in the Great Lakes Basin. And our lab over the last several years, and I guess it's almost 20 years, has been really working hard to focus on improving the fitness of offspring for reintroduction efforts. And the four focal species we spent a lot of time on over the last little while has been specifically, you can see in the top left, red-side dace, lake sturgeon in the top right, Atlantic salmon bottom left, and of course, bloaters in the bottom right corner. And for the purposes of today's lecture, I'm going to keep it relatively light. If you want to get some details, you can certainly grab publications for our website, which I'll point to later. But today, the focus of today's talk will really just to remind people, probably on this call, I don't need to spend much time on it, so I've only kept it to one slide, that there really is a freshwater biodiversity crisis. So I'll just point to that point. I'll talk about what is reintroduction biology for those of you that may not be as familiar. And then I'll go through 3 case studies related to reintroduction, the Atlantic salmon, followed by red-side dace. And I left the bloaters to the end to keep the core going to people on the call to the end. It's like what they do on the radio. They don't tell you the answer till you stay longer. So I got to keep the bloater people on. And then I'll stop by having one slide about my takes on hatcheries. I am a big hatchery person, work with all the different hatcheries I can, and they do incredibly important work that I don't think is recognized enough. And I'm happy to work with them to try to improve the fitness of fish for conservation purposes. So jumping into the talk, really, everybody here on the call is probably well aware that, freshwater biodiversity is in crisis. This is a paper you may not have seen, which is why I put it up. It came out only a couple of weeks ago in the journal Nature, indicating that 1/4 of freshwater fauna is threatened with extinction. So this is not just fishes, but other species of freshwater biodiversity. And the paper, if you actually go read it, will sort of talk about the idea that we don't have good proxies for measuring this. And some of the measures we've done before are probably underestimating the crisis we're in. So on the bottom, you see all the usual characters of threats: habitat destruction, pollution, climate change, invasive species, and of course, overharvesting. These are not exhaustive lists, but the review identifies these as some of the major ones. So normally the group, I would talk more about this, but I think this group doesn't need more lesson about biodiversity loss in the Great Lakes or in freshwater in general. So what I'll do then is I'll sort of shift gears and say, if we want to do something about it, there's a lot of different strategies. We need to do habitat restoration. That's a number one thing. But a lot of times we've done habitat restoration for over a decade in some of these systems, but we don't see the biodiversity coming back. And so what was proposed a few years ago in 2014 is a very influential review suggesting that maybe, and I want to be clear here and say maybe, reintroduction biology could be a tool in some circumstances to help biodiversity. I'll preface this by saying that we really don't want to do biodiversity. If we can avoid it, it's quite expensive, it's quite costly, but in some cases, it might be a necessity. So if we look at the field of reintroduction biology, it's actually an incredibly young field with really the first two textbooks, formalized textbooks, coming out in 2015 on your left from Australia and New Zealand, and 2016 on the right for a more generalized approach to wildlife. And so you can imagine being in this field is kind of an exciting place to be because there isn't a lot known, and a lot of this work is, changing quite rapidly. So I found myself in this space, and I find myself really learning as I go, because there isn't a set in stone kind of approach to reintroduction biology. So if you're asking yourself, what is reintroduction biology? Many of you on the line probably have an idea about it, but I'll just kind of go over it generally speaking. So reintroduction biology, you might know, is the process of releasing a species back to where it was historically, but it became extirpated for whatever reason. And so the ultimate goal in the middle point says that it's to reestablish a self-sustaining population with limited or no intervention eventually. So depending on the species' life history, the idea is that you want to give it a helping hand. You may have several waves of reintroduction, but the goal eventually is to step away and hopefully have it become self-sustaining. But this planning might take decades for species like sturgeon, for example, because of their life history. And the last bullet says that reintroduction biology, to be quite frank, is really considered radical in the sense that it pushes the line backwards by bringing a species back to the landscape in some cases where it's been gone for decades. And so reintroduction biology, we don't take it lightly, and it's something we have to really think about carefully before we decide to even do it. So what does it look like? So what does reintroduction biology look like? So this is a sort of an overview of what it looks like in a general sense. It can be applied to almost any species. And what I'll do is I'll sort of spend a few minutes here to sort of get the idea down. So what we have here, and I don't know if my cursor will show up on the screen or not, but I'll try it. So on the far left on your y-axis, you've got population size from small on the bottom to large on the top. And then you've got the three major phases, which includes what's happening in the wild prior to reintroduction, what happens in captivity in the middle, and then what the reintroduction looks like on the far right side. And on the x-axis, you've got that time across the bottom. And that time, like I said, for some species like sturgeon might be 100 years. For other species, it might be 40 years. So it's really all relative. And then you'll see the other feature is that on the far left in the wild category, you see that initially we have a case where this dashed line represents wild animals, and you see a precipitous decline in the population. So some kind of stressor is impacting the population, and as a consequence, the population goes way down. So you can see the population crashing at some point. So when this happens in the wild due to all these stressors, what ends up happening is we decide at some point to bring in animals to create a founding captive population. And in brackets underneath there, if you're following along, it says source population with an S in brackets. And that's the most important brackets I've ever dealt with in science, because we often don't know, is it one population we need, or should we source several populations when we're doing reintroduction work? So then you get into the captive setting, and you can see in the middle here that the line goes dark black because that's a captive situation. And what we generally have in the captive situation is we have some kind of stochastic fluctuation where we start figuring out how can we raise these animals. A lot of times animals like bloaters or other species we're going to talk about today, nobody has a lot of experience raising them, historically speaking. So we often have this stochastic fluctuation in terms of how well it works. But if all goes well, eventually you want to grow a captive population to some kind of secure size in a captive setting, obviously, in this case, generally a hatchery. I mean, I happen to work with zoos and aquariums on this as well. And at some point, we also have to manage that captive population because it's quite an endeavor to hold on to that population. So I'm currently working on projects, for example, where they have some living animals and some cryopreserved sperm for the genetics. So there's different ways to manage this captive population. But ultimately, the goal is to have an increased number of individuals in captivity so that we can move on to the reintroduction phase, where we're going to essentially select individuals to essentially be part of that reintroduction. And if you're following along on the image, you can see that there's multiple arrows pointing down. And so those are animals from captivity, hence the dark line, multiple waves of reintroduction being done over the same season, over several years, or in the case of sturgeon, maybe for a few decades. And so the ultimate goal, of course, is to move away from that and then have the population in the wild, that dashed line now, become secure and self-sustaining, and then, of course, rise up in terms of population size. It may never get back to the original size of the population, but that is ultimately the goal. And then we have to manage that reintroduced population over time. So lots of different stakeholders, lots of agency, lots of issues in terms of relationship building to manage these issues. But holistically, this is what a reintroduction looks like. So what I'm going to do now that I've given you a quick overview of what the reintroduction looks like, we're just going to do a couple of case studies, and each case study is quite different. So I wanted to give you different perspectives on the challenges we face. So the first one you might be very aware of is one that was essentially initiated several years ago, where from the Canadian side and the American side, there was a real effort to restore or reintroduce Atlantic salmon to Lake Ontario. And you might be very familiar with this story, but you can see here this picture, which is taken by John Clayton, a colleague of mine. You can see the Chinook salmon in the front and the little old Atlantic salmon trying to follow up behind. And I really like this picture to open the story because this picture really shows the challenge of reintroduction, because you have a species that's economically important in the Chinook salmon, and then you have the Atlantic salmon, of course, that's being outcompeted to some extent and has less numbers. So this is a kind of situation where it's a very challenging effort to manage these different interests. So a little background I like to talk about, because I'm a big fan of hatcheries, is that, Atlantic salmon were essentially on the brink in the early 1900s, late 1800s, and there was already some historical attempts to restore them back to the wild. And so back in the day, they had this Wilmot hatchery. You can see kind of a cool picture of it there. And the idea is that they did yearly stocking of eggs, fry, and yearling, even back in that day. And they had some early success based on the records that we read, but it essentially succumbed to the eventual decline. And that decline eventually was caused by presumably overfishing and habitat pressures. We don't have good data for it, but we have good historical records suggesting that might be the case. And eventually they were declared technically extirpated in 1898. And so that is an important point that'll come up later on. So from the Canadian side of things, on the top of the map, the good old north side, we have Lake Ontario here. And as you may be aware, of course, Atlantic salmon were an abundant top predator. We weren't sure if this population originally was landlocked or anadromous. It wasn't super clear based on what we know. There's different indications of what was going on. And the important part of number 3 on the bottom there, it says that a source population does not exist. And this makes it a unique challenge for Atlantic salmon because we can't go draw from a similar genetic population because it simply doesn't exist. So what did we do instead? Well, there was a huge number of partners. So, this is just a little bit of data I'm showing with some colleagues from Ministry of Natural Resources, but tons of partners were participating in this program from all walks of life, whether it was a power generation contributing, whether it was OFA, which is our angling group, or whether it was scientists like myself. So there's a large numbers that worked together, which was an interesting thing. And essentially what they did is they focused originally on fish production and stocking. So there was a large effort to produce enough fish for reintroduction efforts. There was, of course, a lot of habitat enhancement that was done across different streams of Lake Ontario. And of course, there's lots of programming going on in the area on education and outreach, which I think is really important, whether it's the classroom incubators that were provided or other things. And then last but not least, where I come in is we did a lot of research and assessment over about 17 years. But in the first 10 years, we really focused on a few key rivers. We included the Credit River, the Duffins Creek, and Cobourg Creek as ways to monitor potential success or not. So one of the big challenges, as I mentioned earlier, is that we don't have a source population back to the original population. So we had to come up with an idea about how we're going to tackle this. So from a scientific standpoint, we met a large number of stakeholders and scientists, and we said, we really have 3 options. We can either identify and introduce the closest surviving genetic relatives. So we'd have to use genetic similarity as a metric. Number two, we could also say, well, let's not worry about genetics too much. Let's find a Atlantic salmon strain that has ecological suitability. That is, say, it's essentially residing in an environment similar to the Great Lakes. And number three says, well, maybe the last ditch effort is actually to introduce multiple strains with different phenotypic traits and different life histories and take more of an experimental approach to see which of the strains would be more successful. And if you've been following along, as many of you may have, you probably know that we did the last one. We essentially, the large number of people ended up stocking out several strains of these populations. So here's a simple graphic showing sort of an overview, and it's very simplistic, but I'll just show you the general overview, is that we have the Ontario population shown on the far left with some ideas about what they were like. So they obviously had a freshwater life cycle. We don't know if they were anadromous, but we think they had the capacity, and they lived during an oligotrophic time for Lake Ontario. Then if you follow on the right, you can see these are the three strains that we ended up using, or three populations, depending on how you prefer to call them. So we had Le Havre from Nova Scotia, we had Sebago from Maine, and we had Lac Saint Jean from Quebec. And as you can see based on the matrix, there were different attributes about those populations that either were similar or not similar to the situation. So some were more ecologically similar, some might be more genetically similar, and other facets we're not exactly sure. So they vary in terms of their life histories. So we end up using those three strains. And what we did that I think was relatively strong things, we worked across a large number of groups and agencies funded by NSERC on the Canadian side, and the University of Windsor, Western, University of Quebec and Montreal, and most importantly, our colleagues at the Ontario Ministry of Natural Resources. We all worked together. And what we did is we really focused initially in the first 10 years on determining ecological and genetic differences amongst the strains that would try to help us decide which one's more suitable. And so I'm not going to go into all the details. We probably had 40 or 50 papers come out of this. But these papers, unlike some of the other ones, were really management driven in the sense that we answered specific questions about the merits of each of these. So I'll just give you an example. At the top, one of my graduate students looked at the idea of whether or not if we stocked out multiple strains, what's going to happen eventually is you're going to have hybridization between populations. And is this going to be a big problem or not? So Chris Wilson and I, Dr. Chris, as we call him on the Ontario side, sort of looked at this and found that we didn't see a large detriment in the first generation. So, just gave us some information about what the consequences may be of having hybrids. Then we had behavioral issues. Then we looked at genetics, and there's a whole suite of papers that arose from this. The main point is that we really were able to characterize the ecological and genetic differences in both a captive setting and semi-wild setting as well. Now, if I show you some of my colleague Chris Wilson's data that we've been working on for quite some time now, I think this is some of the most poignant kind of endpoint data for this work. And this is work that's coming out still. And this is the idea that we did a match plant introduction. So this obviously means that we reintroduce at the same time the different ages. So essentially at spring par, fall fingerling, and yearling. So imagine, in the sense of fish world, it might be essentially kids from, elementary school, some kids from high school, and some kids from university. So different ages, of course. And I give these talks to non-fish people who don't know this kind of thing. But essentially, we had these three different age classes. And what was really unique and what Dr. Chris really brings to the table is that he's able to track, genetically speaking, right back to the strain and even back to the parents of those offspring. So by being able to do this, we can assess the optimal life strain and stage of life that is working in terms of reintroduction. So let me just show you some of the data that Chris collected over the years. So what we have here on the top, you can see these are the strain origins of the sampled adults. On the y-axis are the number of adults where we got samples from the river. And essentially, without going into too much detail, you can see the different colors. And I'll just walk through the colors. So the green is ambiguous, meaning that genetics could not differentiate the source population. And then we have sort of a darker red is New York Sebago, so coming across the lake. Yellow is Lac Saint Jean. The Sebago is from Ontario is the lighter red, and then Le Havre is the strain that's blue. There's a lot going on in this graph, and I think, I can just sort of summarize by telling you that there's a few things you can see is that the number of adults, this is for a particular river, changed over time. These are the rivers we talked about earlier. 2007, if you see that one little guy on the bottom left, the blue one, that's an interesting backstory. I accidentally caught that Atlantic salmon while I was working on Chinook, and I didn't actually know it was an Atlantic. Kind of embarrassing to tell you that. But I showed it to Dr. Chris, and he said, I hope you got a fin clip. And I said, I did, because I'm still like that. So anyways, that 2007 fish has always been special to me, that little bar. But what you can really take home from this graph is that there's variability in terms of which strains were more successful across the years for adult returns as the proxy. And what you can see is quite a reflection. The main thing you probably notice is you don't see a lot of yellow. So even though the Lac Saint Jean came in at a different time, the amount of yellow was relatively low. So one of the good things about this is it sort of suggests that maybe Lac Saint Jean is not as ideal as a source population. If we move into sort of looking at what life stage is the optimal one, if you look at the Credit River, Le Havre adults, if you look at which ones came back, what you can see is a lot of variability again. So if you look at the y-axis, the number of adults sampled, and the x-axis is looking at the different years, you can see essentially if you go down to the bottom of the graph, the red is the yearlings, the yellow is the false fingerlings, and the blue is the spring par. And what you can see essentially is that maybe stocking out the fish earlier may be more optimal. So you sort of got an idea that the strains differ. And here you can see that, of course, the different age classes also differ to some extent. There's a lot more going on than I can speak about on this graph right now, but that just gives you an idea of the power of the genetics that Dr. Chris Wilson allows because he's done this pedigree work. And I think some of the most important outcomes for me personally are that, that we've essentially mobilized this knowledge. We work with our partners, in this case, the fish culture folks, and we essentially were able to transmit that knowledge, and they were very responsive and receptive to listening about the information, and they essentially started working more on a couple of the strains than the others. So Lac Saint Jean was sort of phased out over time, and they sort of made decisions based on the best available data at that time. So I think this is a good exercise in terms of working with partners and showing that we can make a difference in terms of choosing source populations and stages of release that make sense. So that's sort of the story for Atlantic salmon. So let me switch gears here and sort of show you a non-game fish for a second. So this is case study number 2, and this one is about red-side dace. And the red-side dace is an interesting one. I'll just kind of glance over some of the details. The gist of it is they live around the Toronto area amongst others. And if you're not familiar with the Greater Toronto area, there's almost 7 million people in the Greater Toronto area. And because of a road widening project in a particular area, they funded some of this research for me over 5 years to examine what is the feasibility of doing reintroduction for red-side dace. So what I did is after consultation with a lot of colleagues, I came up with a game plan that looked like this. And it sort of reminds you a little bit about the Atlantic salmon. At the top, we needed to confirm, are there ideal translocation or reintroduction locations? We need to identify a donor population. Where can we get fish for this? And then essentially where one of the big juggernauts are these red dotted lines. We had to see whether or not allowable harm, which means are we going to do more damage to the source population than we would think is okay? Is this going to be exceeded? And because depending on that decision, we'd either go to the right-hand side of this figure or the left-hand side of the figure. What I can tell you without too much detail, and this is all in the publications we have out there, is that we ended up going to the right-hand side because we didn't feel there was a good donor population to have. So I'm just showing you that big red box on the right is suggesting that allowable harm was going to be exceeded in our opinion. So we started studying facets on the right-hand side about captive breeding, reintroduction, and as we mentioned earlier from Bo, transport stress for red-side dace. So what we did is we said we've got to do some homework first. So what we started doing for this reintroduction feasibility study is we surveyed the sites, we began captive breeding, we examined a few of the stressors to decide what suitable locations might be for stocking. And I'll just give you a quick overview on one of the main ones we figured out, which was, of course, thermal stress. So we all know that surveying sites with species at risk is not easy. Some colleagues in the Mandrake lab have used videos to try to do this, and that's been relatively effective, but quite time consuming. So they're using machine learning now to try to optimize the video analysis. But what we did instead is we worked closely, myself and Dr. Chris Wilson again, worked closely on using eDNA, so environmental DNA, to survey all the different rivers and streams across the range we were working in. So this is one of my graduate students, Ryland. And Ryland, essentially, as you can see there in the background, there's that yellow thing on the ground. He was essentially bringing a sledgehammer to these different rivers to essentially break through the ice and grab water samples during the winter because we were trying to compare winter range and summer range for the red-side dace. And if you've never done eDNA, which many of us, most of us have seen this before. Essentially, all you do is you take the water sample, collect it very carefully, avoiding contamination, and you pour it through essentially a relatively inexpensive filter system. And after you pour the water through the filter and have it funneled through it and through a vacuum, you then take that filter and carefully fold it up and then put it in a preservative and bring it back to the lab for analysis. And so what the end game here is, once you get that water sampling done for all the different streams and rivers, you get something like this. This is just one of our many, many maps we have. And what you end up getting is sort of a continuous variation, so a measure of whether or not you detect or don't detect red-side dace in those areas. So this is just one tributary that we did amongst many, many tributaries. And you can see in the top right, there's a legend suggesting that either you go from no detection up to 5 copies per reaction. So that's just a function of the genetics. And so what I think the value of eDNA is in this case is it sort of allows us to triage all the different locations and then send out our teams to the ones where we get at least some detection. So I don't think eDNA is the end all be all, but it certainly helps optimize some of our work. So the next stage is we needed to create a captive breeding program. So we did this at the facility I started here in the Windsor area called the Freshwater Restoration Ecology Center. And I think one of the clever ideas we all had over here is that we said, you know what, we can't work with these species in the wild because they're endangered in most places. So we said, is there somewhere they're not endangered? And it came to us that they're not endangered in the southern parts or middle parts of Ohio. So we went down and grabbed several hundred fish from Ohio with our colleagues down there, brought them back and began a captive breeding program. And after 4 painful years of graduate student work, we essentially had our first success. You can see my fat fingers down there and my thumb squeezing a red-side dace female, producing the eggs. And the picture on the right now is showing you the successful fertilization and ultimate hatching of these animals. So I can tell you now, that's the first time I've ever seen tears in grad students' eyes when they had a fish actually produce eggs like this. So that was a big effort over 4 years, and we have some ability now to produce, quantitatively speaking, enough fish that might be viable for reintroduction. And last but not least, of course, I mentioned that we have to know, where can we actually put these fish out for reintroduction? We need to understand the stresses. So some of the stresses we're dealing with is we're looking at non-native species in terms of predation. So are there brown trout that are just going to eat the red-side dace? How much turbidity is in the stream? We also learned that artificial light, can be quite problematic because this is an urbanized population. And of course, thermal stress. Thermal stress is referring to the idea that many of these streams are in urbanized areas where the water hits the pavement and then ends up in the streams and can essentially change the water temperature by 4 or 5 or even 6 degrees quite rapidly in the summer. So just focusing on all the different stressors wouldn't be viable for this talk. So I'm just going to focus you in on thermal stress for a couple of minutes. So again, all credit due to my postdocs and grad students. This is Andy Turco, one of my postdocs who now is a professor at the University of Guelph. And Andy essentially went streamside and essentially worked up, this is for the Ohio population, but eventually we collected other data, but he worked up a streamside system where he could essentially ask the question, what is the thermal tolerance of the red-side dace? And if you've done this kind of work before, we often have a fancy word for it. We just call it, what is the CT max or critical thermal maximum tolerance do they have? So what you're looking at now is you're looking down at a water bath with 5 different fish, one in each of the containers. And what we do is look essentially, where do they lose equilibrium in light of the temperature? And that's what we call CT max. So let me just show you just a snippet of the data. Again, there's lots of publications out on this from our lab. So Andy did all this great work with a couple of my grad students. And what you can see here is a data visualization for just some of the data he collected. He did over an entire year looking at different seasons. And what he found, if you look at the y-axis, is he looked at the CT max, which ranges from 25 to 35 degrees Celsius. And on the bottom, you can see just a subset of his data, is that you get seasonal fluctuations in CT max. May not be that surprising, but then you do see subtle differences between juveniles' ability to tolerate and adults' ability to tolerate. So these are sort of important considerations. Likewise, Andy also did experiments where he altered the diets of the animals. So you can see on the left, he actually made some animals a little bit fatter and some animals a little bit skinnier based on assumptions from the wild for food availability. And on the right-hand side, the consequence of this that I think is really important for stakeholders out there, or especially practitioners, is that if you can restore habitat to a level where lots of insects are coming back and the fish have access to a lot of food and they have a higher condition factor, they're able to tolerate a few more degrees of thermal stress. And that may mean the difference between life and death for red-side dace. So all these people doing habitat restoration or planning for it, it's super important because it increases the condition factor and as a consequence, increases survival of these animals. And last but not least, of course, we also took all this data and much more of it that we collected, and we started making a plan of how we would use this information to plan out optimization of source population selection across red side dace. There tends to be three genetic regions. And so geographically speaking, how do we manage that? Much like the Atlantic salmon story we talked about earlier. All right, now to keep the core going and people happy, we also worked on the effort with our colleagues at Ministry of Natural Resources and Ontario Fish Culture and our colleagues at USGS and others, US Fish and Wildlife. And a few years ago, you might remember that Ontario was quite heavy into the bloaters. They still have some stocks. And so I was brought in early on to look at the differences between the life history of hatchery and wild-based fish. And so as you can see in this cover picture, you can see the typical hatchery fish at the top and the wild progenitor on the bottom. The one at the top is 2 to 3 years old, and the one on the bottom, I think, was about 5 or 6 based on my memory. And so you can see quite radical differences in their shape, size, and life history, even just from this picture. And the idea is there sort of a mismatch that may be happening in captivity? So we started looking at these questions. So the reason this came up and the reason I was brought in to collaborate is because there was a challenge. And that challenge was that there was relatively low hatching success for the hatchery-based bloaters, initially anyways. And so what they had is a situation where you can see here in the graph on the y-axis is the eyed eggs rate up to percentage. And on the legend, you can see the black is the wild and the open or grayish bar is the hatchery. And so when they had eggs coming straight from the wild, their hatching success was relatively high. But when the eggs were coming from the hatchery, the hatching success, at least early on in the program, they certainly increased the quality over time, but the hatchery process was relatively less early on. Likewise, a couple of other metrics they followed is they looked at survival to 12 weeks post-hatch, and the same kind of story holds true. Likewise, if you look at the cumulative survival to rearing tanks, it's the same general story. So there's something going on where the wild and the hatchery are different, and the question is why? Why is there a big difference? So what our team did is we collaborated with a bunch of people that were able to get us wild fish and hatchery fish, and we took sort of a 3-step approach. So we wanted to ask, why is there poor hatching success? Is it partly because of the differences in the phenotype or what was going on? So the first thing we did is we characterized the wild caught bloater life history we were given. So these are fish provided to us by US Fish and Wildlife and USGS. We then compared wild versus hatchery eggs. And then finally, we made some recommendations about what we thought was driving these differences that we saw. So let me just show you a few figures here for fun. Now, this is arising in a publication from one of my grad students that's she got a job early on and another one took a PhD position. So it got stalled, but this one will be out shortly and I can send you an early copy if you want. But what we have is we had data for about 98 mature wild individuals, and their approximate age based on the otolith work done by a colleague from US Fish and Wildlife was approximately 8.5 years with a range of 5 to 14 years. And as you may know, especially I think I saw Gordon on the call, you can see, of course, that when they haul them up, there's incredible barometric pressure. So there's a lot of gamete loss. Essentially, eggs are being squeezed out of the females as they pull them up from the bottom. And that point becomes very important in a couple slides. So we sort of have a characterized history here for the females, and you'll see how this contrasts with the hatchery females. So to do this comparison, and this is what shows up in the publication, we had 45 hatchery origin fish from 2012, 2013 broods, and we had 43 wild origin fish that had barometric issues. Remember, they got squeezed pretty hard on the way up. And so we compared some basic things like fish length and weight, GSI, condition factor, all the usual metrics. But then we took it a step further and started looking at very specific egg-related metrics. So we looked at egg size, content. Importantly, we also looked at carotenoids and fatty acids, which is relatively unique. So this is comparing, again, hatchery and wild-based fish. So let me just show you a little bit of this data. So I know I'm kind of zipping through here, but I'm just going to show you a little bit of data. So what we have here is the body mass of the mature fish. So these are sexually mature fish, excuse me. And on the left, you've got the hatchery-based fish from 2012, 2013. So they're known ages. And on the right, we know that they're wild fish, but they vary in age. But you can see that the mass at sexual maturation is radically different. So the wild fish are much smaller when they hit sexual maturation than are the hatchery fish. Maybe not that surprising. Then we looked at condition factor. We sort of saw that the hatchery fish were sort of very high condition, meaning they had really access to good foods and fats and things like that compared to the wilds. We see another big difference. And then I think this is a big one here. So this is that issue of what's called GSI. I'm sure most of you are familiar with GSI, which is that gonatosomatic index. So the percentage of their body weight represented by eggs. And the important caveat here is remember that the females are being essentially oozing eggs as they're being pulled up. And despite the fact that they estimate they lose about 30 to 40% of their eggs on the way up, maybe even more, the females from the wild had much higher GSI. It shows equal here, but that's after the fact, after the barometric pressure. So you can see even after barometric pressure, you can see that the females likely have a much higher GSI from the wild compared to the two hatchery stocks on the left. We looked at egg mass amongst them. There wasn't a huge difference, no significant difference, although it was trending towards it because the coefficient of variation around the eggs was relatively small. So the wild eggs were a little bit bigger. But I think what more importantly, oops, my screen is doing something funny, sorry. So what may be more important though, is the fact that when we looked at the carotenoid content as an indicator of quality rather than just size, we saw a radical difference in the amount of carotenoids in the diet. So the fish in the wild had a massive, massive difference in carotenoids, much higher. And as many of you probably know, that carotenoids quench free radicals. So these are things like oxidative species that are essentially bad for the eggs in terms of hatching success. And the carotenoids do this in a sense that it helps the juveniles survive the hatching process. So seeing that difference in carotenoids was very enlightening for us. Likewise, we had a panel of fatty acids run by a colleague of mine in Toronto, and essentially he looked at a whole suite of fatty acids for us. This is just a summary here. And you can see in the top there, you've got the fatty acids in the far left, you got the hatchery eggs, and then you got the wild eggs. And I'll just show you that in some of the examples, you can see at the very top, there's a six-fold or almost seven-fold difference in certain fatty acids, which are important for hatching success. Twice the difference for the second one, which is responsible for immune differences. DHA has been correlated with brain and things like that. That one wasn't that different, but some of the other panels on the bottom were quite different. The PUFA ones, which are very commonly assessed, were quite different. So again, we learned a lot from this assessment of the eggs in the hatchery and in the wild fish. So our real take on this is we made a recommendation saying that, there might be an issue here in terms of the life history in the hatchery. They're obviously growing much bigger and different, maturation time. We said maybe you need to consider a custom diet to deal with the carotenoid issue. We worked with the ministry on using LHRHA and carpituitary extract, mostly LHRHA, but also carpituitary extract to essentially improve egg extraction. We made the suggestion maybe you need to use older broodstock that have been grown longer and more mature. And on the bottom, it says that we're really interested in the fish culture system, whether it's Ontario, Nova Scotia, British Columbia, United States, doesn't matter, about this environmental mismatch that may be happening in the hatchery. So what happens in a hatchery sometimes, and again, it depends if you're making fish for put and take fisheries, different, but if you're trying to make conservation fish from a hatchery, sometimes you end up with this environmental mismatch. And so in this graph, you can see there's a blue distribution of traits that are seen in the hatchery fish, and the green bar on the right is the optimized phenotype or body shape or physiology or behavior that's better in the wild. And so sometimes if you produce a mismatch like this, it can be problematic for the fish once they've released into the environment. So our lab, as Bo identified at the start, have always been trying to minimize that environmental mismatch to essentially promote the fitness of fish being produced for conservation purposes. And so this is something we've been fortunate enough to work with our colleagues here in Ontario. So I'm just going to wrap up. I think I'm on time here. So I think I'm going to wrap up with what I call my hot takes. And if you don't know the term hot takes, it just means kind of controversial ideas. So I just like to bring these up. I am the biggest hatchery person you'll ever know. I go to hatchery. I work with a lot of colleagues, just always trying to learn from them. And so what I've learned from talking to all my colleagues in the hatcheries is that the environmental mismatch is often common because it's hard to replicate a wild environment in the hatchery. It's obvious to all of us. And it's often very difficult to do it because things like enrichment, which I've been testing for a long time, are not easy to clean in the hatchery. They're not easy to manage. So it costs a lot more to do it. So I've learned a lot from my friends. What I will say in terms of my fish hatchery hot takes is that hatchery practices have not really evolved. And I use that lightly, that term evolved as a pun. But the idea is that many of the hatchery practices haven't changed for 60, 70, 80, 90, maybe even 100 years. So this is why I think I've got a great research program in terms of collaborating with many of you, because I can learn from you and maybe you can learn a little from me. I think genetic diversity its focus on genetic diversity is completely misguided. I won't go too much into this, but I'll tell you that the original focus on genetic diversity arose from zoos where they were working with the last 12 individuals of species. And when they managed that, people adopted that framework for hatcheries, and that was a huge mistake. So I think genetic diversity, there's too much focus on it, and we need to think more about adaptability, genetic adaptability, not just diversity. The next one says natural selection and sexual selection are completely ignored. The picture on the left is your typical situation where there's a bucket of milk poured into a bucket of eggs. And in that one swirl of the bucket, you just wiped out, 150,000 years of natural selection and sexual selection in one swirl. So that's really been an issue where I think we can improve some of the fitness of the animals. And last but not least, again, I think the real critical issue for some of the fish hatcheries around the world is that their performance indicator is often the number of fish they produce, but that's always a different metric for conservation. For put and take fisheries, number of fish makes sense. For conservation, though, I would say we need to focus more on the quality of the fish. That is to say, the fitness of the fish, potential fitness, rather than just the number. I mean, I know the number is easier to measure, but really, I would rather raise less fish that are more environmentally matched than a large number of fish that are mismatched. So those are my controversial hot takes to end the story. And with that, I just want to make sure that I say that, reintroduction biology, of course, as I mentioned, is becoming more common. So we need to know more about it. Choosing source populations is very complex. I kind of went through a lot today, but it's very difficult to do. There's lots to learn about reintroduction. We don't know a lot, and we're really learning quickly. I think hatchery practices need to evolve a little bit. That's not a criticism, but just saying we need to collaborate more together. And I glossed over a lot of detail today, but all the publications with incredible detail can be found on our website, which is located there on the bottom. So I know it's hard online to take all that in, but I appreciate your time today. And it was a pleasure to kind of go over some of the research my grad students, postdocs, and a lot of my collaborators have worked with me on for the last couple of decades. So I'll be happy to take questions, Bo, at this point.
Yeah, fantastic. Thanks very much, Trevor. Great presentation, a great sort of flavor of all the breadth and scope that you and your students and postdocs do. And I do hope it's generating a lot of questions. You can, ideally, you can just throw your hand up throw your hand up like Eric's done there. Throw your camera on to be a little bit more personal. Ask directly. If you're not comfortable with that, you can also put them in the chat. So, Eric, wow, fast question. Go for it.
If I get the camera on, we'll see. One question I did have is what I didn't hear, I guess, actually, in the presentation. I didn't hear anything about First Nations or tribes being incorporated or being worked with throughout this whole project.
Yeah, so it wasn't so much an oversight. So I'm actually right now, I'm working with here on our side of the border, Walpole First Island Nation. And we actually are building literally right now. My students are there today. In fact, they're building a fish hatchery at the school. It's modeled after the Atlantic Salmon Program from Toronto, where they're going to have classroom incubators. And they're going to do ceremonial releases in the next 5 years of lake sturgeon. So they're focused on lake sturgeon. So I didn't bring it in because I didn't feel it was my place to tell you about their programming, but we're supporting their programming right now. And we consult with them on some of these reintroduction efforts. For example, at the Red Side Days, this is one where we're working with our Department of Fisheries and Oceans to collaborate with our Indigenous partners in Toronto. But it becomes, like I said, a large number of stakeholders and Indigenous colleagues. But going back to the main point, I think you're right. There's an important place for them in the story. And they've engaged us over the last 8 years. And this has been an exciting time for us because building a hatchery at their school has been really fun, to be honest with you.
Thank you. We just started reintroduction of lake sturgeon in the Boardmanoway rivers and are using streamside rearing with flow-through systems. So
yeah, and so this was meant to be a community effort. So they are doing the same kind of thing at the school because they're right on the waters there. As you might know where Walpole is. So it's called Bekejinon. So Bekejinon First Nation.
Thanks, Eric. So while we wait for another question, maybe we'll go back. I have bloater questions, but I want to go back to Atlantic salmon. So really curious about that lake. I mean, really cool that earlier the par seemed to perform better and you get more adults out of the par, which is really interesting and sort of maybe some of the signal we're seeing with Saginaw Bay Cisco reintroduction. We're doing like spring. I mean, 40 millimeter versus
Mhm.
like fall. And we're seeing better returns of the smaller ones so far, which I don't think we would have expected, but you're seeing it also with Atlantic salmon. For Lake St. John, I don't know if I'm saying that right, but the strain of the population that really didn't perform very well. Do you all have an idea of why that strain did not perform well so far or is being phased out in Lake Ontario?
Yeah, so two parts to your question. The first part I think is that one possible correlate that's happening is that it's possible that this younger fish stocked out earlier is better, partly because they become less domesticated and that environmental mismatch has been minimized by time. And that's something we've been looking into. So that may be part of the answer to your first part. When it comes to Lac Saint Jean, there's sort of two things going on there. One, they weren't really successful in the husbandry space. They were hard to rear in the facility. But also the second part is they just weren't seen out in the wild. So it's kind of a two-part. They weren't doing great in captivity. So when you're stocking them out, they weren't doing great as well. And again, this is a holistic story, bigger than the data I just showed, obviously. But I will say that the answer to that might be that they just weren't adaptable to either husbandry and or the rivers themselves.
Interesting. Okay.
But they were eliminated, by
Uh,
the way. You asked that as well. They were taken out of circulation
yeah.
at some
Okay.
point during the process.
Yeah. Jim, you have a question? Follow up on salmon?
Yeah, Trevor.
Hey,
Um, I'm trying to get my camera to work here. Let's see.
it's all good. Yeah.
There we go. Good to see you.
Yeah,
Thanks for the
I know what you look like, so
presentation.
it's okay.
um, with regard to the genetic comparison of the strains, it sounded like at some point you were comparing it with the original extirpated strain, and I should know this, but were there enough old tissues to be able to do that sort of thing?
No, so there was that caveat about the Lake Ontario one. So there was an interesting article I can share with you later where they actually did go to the Royal Ontario Museum and found one remnant stuffed Atlantic salmon from the original population where they got DNA, but the comparison is very limited by that. So that's why that was a caveat. So those were sort of guesses, best guesses on the far left. So that's why we took the three strain approach, I think, is that we didn't have good information on the original population.
Right. Okay,
But
that
they did
makes
find
sense.
one. It's a fascinating story, by the way. Stuffed, and they did use special techniques called ancient DNA analysis to extract DNA. So kind of a great story.
Cool. Thanks.
By the way, Jim, going back to one thing I didn't say is that one of the things in the hatchery systems too that we all know about but don't think about is that, and you have thought about it, is that there's no olfactory cues in some of these facilities, so they don't match the areas where they're going out. So the environmental mismatch I talked about, we've been studying two facets lately. One, as you know, Jim, is olfactory cues. So oftentimes the hatchery doesn't match where they're going. So is that a problem? And the second one that we started doing recently that I presented just a couple of days ago to my colleagues in Ontario is there's a color mismatch. So we've been testing this idea, and I think I've got a slide here for you guys, if you're willing to let me show you. So here's our system here where we have all these little mini tanks, essentially just fancy buckets, right? But we're raising them in these different colored tanks that emulate many of the different hatchery colors, whether the tank's green, gray, or blue, which is very common. And I don't need a bunch of stats to show you. Here's the same families, exact same eggs, exact same moms and dads. And look at the difference in the top left. You can see that's a white bucket fish. In the middle one is the black. These are the same families of Chinook salmon. So you got the white on the left, black bucket in the middle, and blue bucket on the right. And again, exact same genetics, same environment until they went into that bucket. And you can see 7 days later how drastically different the color is. And we noticed this in Nova Scotia when we were working on reintroduction of Atlantic salmon in Nova Scotia, that they looked really bluish green. And I said, don't the birds see this? They said, yeah, they get hammered by birds. I said, Disney, and they realized their tanks are really, of course, green. And so this is something that they kind of had us start working on because of this problem. So we're doing this with Chinook and Atlantic salmon. So one for the West Coast, one for the East Coast, to see if tank color might be an issue. And the way we might solve this, in case you're interested, is we're now about to, this is happening live. So if I go back, where's the picture? I don't know if I have it, but this is from a week ago, these data. And so what we're going to do is actually line the different buckets with liner that you can buy commercially for hatcheries and put in a picture of kind of like rocks and things and see if we can reverse the color changes over amount of time. We want to see how plastic this response is. But again, this became an issue for the post-release stocking that we noticed in Nova Scotia.
Right. So historically here, Trevor, when we were getting the Atlantic salmon ready to be released into the streams of the St. Regis or wherever we were doing that, they would be put out in the raceways, which are concrete, because they were too blue from the tanks. And after a couple of days, they would look more like that color.
Yes.
What we do now is just we dim the lights a whole lot so it gets dark, and then they darken up.
Agreed 100%. And what I've learned, and this is a great conversation, is that when I visit all the different hatcheries around North America and other parts of the world, such different practice across them, and nobody really shares a lot of this. And so I tend to be a conduit between people for some of these ideas. So I don't take any credit for them. I'm just saying I talk to different people. And what you describe is really good, but the one in Nova Scotia left them in green troughs before they went out for reintroduction. So now they're going to transition them to potentially gray tanks before stocking for at least a week or two.
Right.
So a small change that might make a big difference in survival, because when they first go out, I don't talk about this today, but we also test hard and soft release. They often end up being shocked when they first go in. So if you're really bright blue against the background and birds can see you, you're going to get hammered. So that's something that we're interested in improving the survival both in the hatchery and afterwards.
Yeah, exactly. Makes sense. And thanks again to you and Ali for all the work you did with the olfactory.
Yeah. We unfortunately we broached some of that right at COVID, so we kind of got cut off by that. But we enjoyed our time in New York. Thanks for having us.
Yeah, no problem. Thank you. See you.
Thanks, Jim.
I can't see the chat, by the way, so I apologize if anybody's on the chat. My computer.
Yeah. So Markel Morfitt said, great to hear about all the cool and important work your lab has been doing. Thanks, Trevor. I got one from Peter Heinerman. I think he just sent it to me, maybe by mistake, or maybe because he knew I was facilitating. He has a question. I don't know if you can answer, but I'll ask it anyway. What would be the secure size for reintroduction of a walleye population when growing a captive population? So I don't know if you can speak specifically to walleye or just generally about sort of what you would aim for with a captive population more generally. Might be a tough question.
I think it's a really important question, because when we were doing the Atlantic work and some of the other work with red side dace, one of the biggest challenges is obviously what can the habitat sustain? And that's not easy to actually figure out a lot of times, so part of the issue for us is, I think going back to the question, I'll never be able to answer that exact question for walleye, but what I will say is that in our calculations, our number one challenge was estimating A, habitat quality, and B, what is sustainable for that habitat. So for red side dace, it was really hard, and it still is very hard for us to determine what is good quality, and secondly, how many can be sustained. So I think it really depends on the situation. For walleye, it's very different than what I've done, but there are tools we've developed to do this for different species. So we can always talk offline as well if you like.
Okay, so I don't know if Peter is still on, but you're welcome to
always reach out. That's the goal of doing these kinds of talks, which is nice.
Yeah. So bloater question. So first of all, just a clarification, that carotenoid content, is that in the tissue, like the body, like whole body, or is that just in the eggs themselves?
Yeah, we focused exclusively there on metrics from the eggs.
And that would correlate with some of the ARA and EPA and sort of PUFAs. Would those all be sort of related to that carotenoid, probably, or hard to say?
Correlated, just essentially the radical difference in the diet between what the hatchery is being fed and the wild is responsible for those differences. And I think the key variables there, as you probably saw, is that those differences are related to hatching success. So that's why we were very concerned about it, because those tend to affect the success of the offspring.
Yeah, very dramatic results there. And so there was a study, and it may have been after your study, I'm not sure, and I don't know if you were involved in it, between some folks at Tunison and Fish and Wildlife Service, and I think White Lake, where they did a diet study with bloater, and they tried to see to what extent does it, are there differences?
I'm familiar with it because I was there for that time, but yes, this is why this paper is supposed to come out at the same time, but the student unfortunately got a PhD, which is great.
Yeah.
So I've got to get this one out. You kind of motivated me to get this thing back on the front burner.
Okay. Well, it's a really cool result. And I'd be curious if anybody's on the call that could speak about to what extent the diet that they incorporated in that study was trying to reflect some of the deficiencies that you saw based on.
Well, I know that the diet stuff was based on the commercially available feed from Japan and Canada and the US. So I don't know if they customize a diet, but I think a custom diet might be the answer for some of these issues you're describing. But of course, I'll let people on the call jump in.
Yeah, Jim, you were on that project.
Yeah, real quick. We had down here and in Lamar, we had three different diets, and one was commercial, what you'd expect, regular diet. One was Otahimi, which had a lot more protein from crustaceans, and one was custom made by the folks out in Washington state that have been doing biochemistry kind of analysis. And then up in at White Lake, they did several others. But the upshot was we couldn't tell that there was an actual diet effect in the end. So that and actually what we think we discovered is that the thermal conditions were much more critical. And here at Tunison, of course, it's all one temperature, and they just didn't want to spawn. So we had trouble evaluating after the fact.
Was all one temperature, and now it's not. Now you've got it. So
yeah, that is right. Maybe too late. I don't
John,
know.
John is also on that study. John, you want to add anything else?
Yeah, Jim summarized it pretty well. The biggest hurdle, and I think if anybody tries to repeat a diet study within a hatchery, to see if you get one diet that's maybe more optimal. The biggest challenge to us was the fish to fish variability. It overwhelmed. I mean, if we really want to test, one diet versus another in a hatchery, we're going to have a huge sample size. So, just keep that in mind moving forward. If anything from our study, can be used as some pilot information to guide sample sizes. And also, like Jim said, the big factor was the temperature. If the temperature doesn't drop down real cold in the winter around spawning time, fish won't spawn.
Yeah, the White Lake station, as you know, the diet I was involved with was a White Lake, but they have a natural water source, so they had some success because they have that variability.
Yeah, and we had some success here in Lamar, Pennsylvania, with ours because we got down to around 5 degrees. And just anecdotally, this year, being a colder than average winter, at least a colder winter than we've seen in quite a few years, it does seem our production fish are producing very well this year.
Good to hear.
Thanks, John. In the chat, we got something from Don Schreiner. Great presentation. How much of the adult return rate difference between life stages could be due to the smaller ones imprinting and maybe coming back there and catching them versus the older ones you stock maybe survived but didn't come back and weren't able to capture as easily as adults?
Yeah, so that's I mentioned earlier, alluded to it, that the fish stations, depending on which one you go to, for example, the one these fish were raised at, for the most part, Normandale, don't have they have a bit of a creek water, but not the same signature as the river they're going to. So I think that's part of that environmental mismatch I showed you, that I think olfaction is a big problem in many of these situations. And I've been talking to Andy Dittman on the west coast of North America, who specializes in olfactory issues. And I think that the answer to your question is that it might be the fact that they had the critical development where they got into the river early enough and they may have, caught that right signature to come back, and or the strain rate of these fish is not really well known, at least not in Lake Ontario. So I wouldn't be able to give a concrete example, but I do think the olfactory mismatch is again, I don't think it's any one silver bullet. I think one of the challenges, some people try to find that one silver bullet, but I think if you saw today's talk, it's trying to eliminate the holistic environmental mismatch, not one facet. So we test one facet scientifically because you need to, but I think we need to think about how do we manage the diet? How do we manage olfactory cues? I think even the mate choice stuff that doesn't happen in hatcheries needs to be managed holistically. We need fitter fish. I think that's really the message for conservation work. I want to stress that, not for put and take fisheries.
Yeah, agreed. Carl, nice to see you.
Hi, thank you. I was curious, I've been a little bit involved with the Arctic grayling reintroduction in Michigan, which is getting ready to go. And one of the things we're planning on doing there, using like remote site incubators or floating basket incubators with the idea of using like eyed eggs in the hatchery and then incubating those in the stream and letting them out at sort of a smaller size. I was curious if you've come across anything like that with the work that you've been doing in Canada.
Yeah, so we've planted out even Atlantic salmon in these incubators. So we have, I don't know what kind you're using, but we have these stacked, they're coming from a company in the West Coast, but a stacked incubator where we can put like apartments of eggs that just able to leave themselves. And we hypothesize based on the data we have that they're putting them in as eggs allows them to hit that critical development time for olfactory cues and the environmental change. So we're a big fan of those. The problem, as I mentioned earlier, is that managers are not a big fan because it's expensive to do that stuff.
Okay.
So again, it comes down to the quality versus quantity of them. So we find that our systems work well, but it becomes an issue for the cost efficiency.
Thanks.
Thanks, Carl. There's also some work, Carl, being done with that for whitefish, I believe, by some of the tribes up north. I do see like Rusty on the call. I don't know if we have anybody else from Little Trappers Bay Band or Rusty from Sioux Tribe that wants to talk about some of the egg incubation work they're doing in rivers for whitefish to see if they can bring back the tributary spawning whitefish. But I can try to connect you with some of those people that are doing it.
Yeah, by the way, I'm happy to connect with folks. And I know we have a tight timeline based on your schedule. So I learned as much from anybody I talk to here than anybody else. So it's great to exchange ideas. I'm not far from you either, Carl. So. All right.
Great.
Good. Well, thanks for having me today, Bo.
Yeah. Thanks so much for doing this. If people want to give Trevor sort of the virtual wave through Zoom, we're getting really good feedback on your presentation. This will be recorded and uploaded to our greatlakesciscos.org website in a week or so. And hopefully you'll be able to see all these comments that are coming in, Trevor. But great presentation. And as he said, please reach out to Trevor if you have any questions, follow-up conversations. You can tell he enjoys talking fish and fish hatcheries.
There's our website on the bottom here.
There you go. Yeah. All right. Thanks, everybody. Have a great weekend.
Have a good rest of your day.